A protocell is a self-organized, endogenously ordered, spherical collection of lipids proposed as a stepping stone to the origin of life. A central question in evolution is how simple protocells first arose and how they could differ in reproductive output, thus enabling the accumulation of novel biological emergences over time, i.e., biological evolution. Although a functional protocell has not yet been achieved in a laboratory setting, the goal to understand the process appears well within reach. Overview. Compartmentalization was important in the origins of life. Membranes create enclosed compartments that are separate from the external environment, thus providing the cell with functionally specialized aqueous spaces. Because lipid bilayer of membranes is impermeable to most hydrophilic molecules, the cell must have membrane transport systems that are in charge of import of nutritive molecules as well as export of waste. It is very challenging to construct protocells from molecular assemblies. An important step in this challenge is the achievement of vesicle dynamics that are relevant to cellular functions, such as membrane trafficking and self-reproduction, using amphiphilic molecules. On the primitive Earth, numerous chemical reactions of organic compounds produce the ingredients of life. Of these substances, amphiphilic molecules might be the first player in the evolution from molecular assembly to cellular life. A step from vesicle toward protocell might be to develop self-reproducing vesicles coupled with the metabolic system. Selectivity for compartmentalization. Self-assembled vesicles are essential components of primitive cells. The second law of thermodynamics requires that the universe move in a direction in which disorder increases, yet life is distinguished by its great degree of organization. Therefore, a boundary is needed to separate life processes from non-living matter. The cell membrane is the only cellular structure that is found in all of the cells of all of the organisms on Earth. Researchers Irene A. Chen and Jack W. Sostak, amongst others, demonstrated that simple physiochemical properties of elementary protocells can give rise to essential cellular behaviors, including primitive forms of Darwinian competition and energy storage. Such cooperative interactions between the membrane and encapsulated contents could greatly simplify the transition from replicating molecules to true cells. Furthermore, competition for membrane molecules would favor stabilized membranes, suggesting a selective advantage for the evolution of cross-linked fatty acids and even the phospholipids of today. This microencapsulation allowed for metabolism within the membrane, exchange of small molecules and prevention of passage of large substances across it. The main advantages of encapsulation include increased solubility of the cargo and creating energy in the form of chemical gradient. Energy is thus often said to be stored by cells in the structures of molecules of substances such as carbohydrates, lipids, and proteins, which release energy when chemically combined with oxygen during cellular respiration. Energy gradient. A March 2014 study by NASA's Jet Propulsion Laboratory demonstrated a unique way to study the origins of life: fuel cells. Fuel cells are similar to biological cells in that electrons are also transferred to and from molecules. In both cases, this results in electricity and power. The study states that one important factor was that the Earth provides electrical energy at the seafloor. This energy could have kick-started life and could have sustained life after it arose. Now, we have a way of testing different materials and environments that could have helped life arise not just on Earth, but possibly on Mars, Europa and other places in the solar system. Vesicles and micelles. When phospholipids are placed in water, the molecules spontaneously arrange such that the tails are shielded from the water, resulting in the formation of membrane structures such as bilayers, vesicles, and micelles. In modern cells, vesicles are involved in metabolism, transport, buoyancy control, and enzyme storage. They can also act as natural chemical reaction chambers. A typical vesicle or micellar in aqueous solution forms an aggregate with the hydrophilic head regions in contact with surrounding solvent, sequestering the hydrophobic single-tail regions in the micellar center. This phase is caused by the packing behavior of single-tail lipids in a bilayer. Although the protocellular self-assembly process that spontaneously form lipid monolayer vesicles and micelles in nature resemble the kinds of primordial vesicles or protocells that might have existed at the beginning of evolution, they are not as sophisticated as the bilayer membranes of today's living organisms. Rather than being made up of phospholipids, however, early membranes may have formed from monolayers or bilayers of fatty acids, which may have formed more readily in a prebiotic environment. Fatty acids have been synthesized in laboratories under a variety of prebiotic conditions and have been found on meteorites, suggesting their natural synthesis in nature. Oleic acid vesicles represent good models of membrane protocells that could have existed in prebiotic times. Electrostatic interactions induced by short, positively charged, hydrophobic peptides containing 7 amino acids in length or fewer can attach RNA to a vesicle membrane, the basic cell membrane. Geothermal ponds and clay scientists have come to conclude that life began in hydrothermal vents in the deep sea, but a 2012 study suggests that inland pools of condensed and cooled geothermal vapor have the ideal characteristics for the origin of life. The conclusion is based mainly on the chemistry of modern cells, where the cytoplasm is rich in potassium, zinc, manganese, and phosphate ions, which are not widespread in marine environments. Such conditions, the researchers argue, are found only where hot hydrothermal fluid brings the ions to the surface, places such as geysers, mudpots, fumaroles and other geothermal features. Within these fuming and bubbling basins, water laden with zinc and manganese ions could have collected, cooled and condensed in shallow pools. Another study in the 1990s showed that montmorillonite clay can help create RNA chains of as many as 50 nucleotides joined together spontaneously into a single RNA molecule. Later, in 2002, it was discovered that by adding montmorillonite to a solution of fatty acid micelles, the clay sped up the rate of vesicle formation 100-fold. Research has shown that some minerals can catalyze the stepwise formation of hydrocarbon tails of fatty acids from hydrogen and carbon monoxide gases that may have been released from hydrothermal vents or geysers. Fatty acids of various lengths are eventually released into the surrounding water, but vesicle formation requires a higher concentration of fatty acids. So it is suggested that protocell formation started at land-bound hydrothermal vents such as geysers, mudpots, fumaroles and other geothermal features where water evaporates and concentrates the solute. Montmorillonite bubbles. Another group suggests that primitive cells might have formed inside inorganic clay microcompartments, which can provide an ideal container for the synthesis and compartmentalization of complex organic molecules. Clay-armored bubbles form naturally when particles of montmorillonite clay collect on the outer surface of air bubbles underwater. This creates a semi-permeable vesicle from materials that are readily available in the environment. The authors remark that montmorillonite is known to serve as a chemical catalyst, encouraging lipids to form membranes and single nucleotides to join into strands of RNA. Primitive reproduction can be envisioned when the clay bubbles burst, releasing the lipid membrane-bound product into the surrounding medium. Membrane transport instead of the more popular phospholipids of modern cells. The membrane of protocells in the RNA world would be composed of fatty acids, and that such membranes have relatively high permeability to ions and small molecules, such as nucleoside monophosphonate, nucleoside diphosphate, and nucleoside triphosphonate, and may withstand millimolar concentrations of Mg2+. Osmotic pressure also plays a significant role in protocell membrane transport. It has been proposed that electroporation resulting from lightning strikes could be a mechanism of natural horizontal gene transfer. Electroporation is the rapid increase in bilayer permeability induced by the application of a large artificial electric field across the membrane. During electroporation in laboratory procedures, the lipid molecules are not chemically altered but simply shift position, opening up a pore that acts as the conductive pathway through the bilayer as it is filled with water. The mechanism is the creation of nanometer-sized water-filled holes in the membrane. Experimentally, electroporation is used to introduce hydrophilic molecules into cells. It is a particularly useful technique for large highly charged molecules such as DNA and RNA, which would never passively diffuse across the hydrophobic bilayer core. Because of this, electroporation is one of the key methods of transfection as well as bacterial transformation. Fusion. Some molecules or particles are too large or too hydrophilic to pass through a lipid bilayer, but can be moved across the cell membrane through fusion or budding of vesicles. This may have eventually led to mechanisms that facilitate movement of molecules to the inside or to release its contents into the extracellular space. Artificial models. Langmuir-Blodgett deposition. Starting with a technique commonly used to deposit molecules on a solid surface, Langmuir-Blodgett deposition. Scientists are able to assemble phospholipid membranes of arbitrary complexity layer by layer. These artificial phospholipid membranes support functional insertion both of purified and of in situ expressed membrane proteins. The technique could help astrobiologists understand how the first living cells originated. G1U protocells are synthetic chemical particles that possess cell-like structure and seem to have some functional living properties. First synthesized in 1963 from simple minerals and basic organics while exposed to sunlight. It is still reported to have some metabolic capabilities, the presence of semi-permeable membrane, amino acids, phospholipids, carbohydrates and RNA-like molecules. However, the nature and properties of the G1U remains to be clarified. In a similar synthesis experiment, a frozen mixture of water, methanol, ammonia and carbon monoxide was exposed to ultraviolet radiation. This combination yielded large amounts of organic material that self-organized to form globules or vesicles when immersed in water. The investigating scientist considered these globules to resemble cell membranes that enclose and concentrate the chemistry of life, separating their interior from the outside world. The globules were between 10 to 40 micrometers, or about the size of red blood cells. Remarkably, the globules fluoresced, or glowed, when exposed to UV light. Absorbing UV and converting it into visible light in this way was considered one possible way of providing energy to a primitive cell. If such globules played a role in the origin of life, the fluorescence could have been a precursor to primitive photosynthesis. Such fluorescence also provides the benefit of acting as a sunscreen, diffusing any damage that otherwise would be inflicted by UV radiation. Such a protective function would have been vital for life on the early Earth, since the ozone layer, which blocks out the sun's most destructive UV rays, did not form until after photosynthetic life began to produce oxygen. Ethics and controversy. Protocell research has created controversy and opposing opinions, including critics of the vague definition of artificial life. The creation of a basic unit of life is the most pressing ethical concern, although the most widespread worry about protocells is their potential threat to human health and the environment through uncontrolled replication.